Hello there, everyone. So good of you to join us today. My name is Catherine Gretzinger, and I'm an associate professor of teaching at the UBC School of Journalism, Writing and Media. I'm delighted to be your moderator for this webinar, Translating Discovery to Transform Health, Untold Stories from Medical Research. This is the third webinar in the Transforming Health for Everyone webinar series, brought to you by the UBC Faculty of Medicine, Development and Alumni Engagement. And just on a personal note, it's been so wonderful to work with people across our university and both campuses, as a matter of fact, over these last several trying months for all of us. So it's wonderful to be able to participate in these important and challenging conversations. For today's event, we are using Zoom's live captioning to ensure that the event is more accessible and inclusive. It may automatically be showing on your screen, so please look to the bottom of your Zoom window for the live transcript button, where you have the option to turn it on or off according to your preference. If you need assistance, or if anyone is experiencing any audio or video issues, you can please reach out to us via the chat in Zoom. We'll be monitoring that throughout and trying to make information available to you. So I'd like to begin by acknowledging, of course, that UBC's Vancouver Point Grey campus is situated on the traditional, ancestral, and unceded territory of the Musqueam people. And UBC Okanagan is situated on the traditional, ancestral, and unceded territory of the Syilx Okanagan Nation. I'd like to recognize that you may be joining us from a variety of places across the province of British Columbia, maybe elsewhere in this land we know as Canada, and maybe elsewhere in the world. All of the territories that we live and learn and play upon belong to someone else before and have been lived upon for millennia and used and thought of in other circumstances and ways. And just important to acknowledge the great privilege that we have, especially at a time of reckoning, when we are remembering and honoring the countless Indigenous children who are lying in unmarked burial sites across the country. These tragedies, along with the circumstance of COVID-19, the very pronounced rise in anti-Asian, anti-Black, anti-Muslim, anti-Indigenous racism in Canada and beyond, present us with very heavy challenges during these times. So we encourage you, and certainly the UBC community encourages you, to reflect on the actions that we can take as individuals and together, collectively, to promote justice, equity, diversity, and inclusion in our decisions and in our daily lives. So today's conversation, we're going to ask you to respect everyone participating in this virtual event. We recognize that we're all coming at this from different backgrounds with different experiences and understandings. Please help us to contribute in creating a respectful and inclusive environment for our speakers and for fellow UBC alumni and community members who will be joining us both live and over time, as this webinar is being recorded and it's going to be shared widely. So for additional information that you may want to get access to, you can visit the links that are being shared in the chat and go and find out more about UBC's Respectful Environment Statement and resources that are available for equity and inclusion services at the University of British Columbia. So let me get now to the task at hand. I'm going to be speaking for a couple of minutes just to provide some orientational conversation, but really the stars of our show are waiting in the wings. And I'm just so excited to present them to you, because translating discovery and helping people to understand how science is affecting people's lives is really a great privilege of working in a university setting. You're constantly surrounded by people who have active imaginations and deep commitments to research and discoveries of truth, and also commitments to helping and shifting and changing the way that we understand the world in which we live. So on behalf of the Faculty of Medicine at UBC, I'd like to extend a warm welcome to everyone joining us today. Whether you're viewing from BC, across Canada, or elsewhere in the world, we're so pleased to be able to present this online webinar during this extraordinary time that we're living through, and we're very grateful to have all of you with us. The global COVID-19 pandemic really catalyzed an unimaginable transformation in how we think about science and healthcare, even how we communicate with one another. As of July 3rd, approximately 68% of the total population of this country has received at least one dose of a COVID-19 vaccine. And now our numbers of fully vaccinated individuals are starting to climb too. Since the onset of this pandemic, the public has been attuned to vaccine development amidst the disruption in our daily lives and the far-reaching impact on health systems and on economies around the world. The three UBC experts that we're so fortunate to have with us today are going to be sharing some of their stories, some of them told, some of them untold, about the long and winding road that scientists have participated in as they've embarked on new discoveries, on thinking about vaccines and drug treatments and tackling viruses, everything from the vaccine the virus that we now know is COVID-19 to Alzheimer's, cancer, diabetes, multiple sclerosis, and many others. These are people who are deeply concerned about finding out what is happening and then trying to intervene in a positive way. BC is home to a thriving life sciences ecosystem that's made pivotal contributions to the COVID-19 response. But what few people realize is that many of the major scientific contributions underpinning the response started right here at UBC. So before I welcome our speakers, if you just take a look at this screen, to help us set the stage for our conversation today, I'd like to refer to a quote from Dermot Kelleher, the Dean of the Faculty of Medicine and the Vice President of Health at UBC. He says that we still have a long way to go, but the pandemic has demonstrated that we can be accomplished at an accelerating pace. As we turn toward recovery, we have an unprecedented opportunity now to invest in BC's biotech sector and accelerate scientific discoveries into new drugs and treatments more effectively and more efficiently for aggressive diseases. So if you think about those words for a moment and think about all that is possible in that, it presents us with not only a challenge, but also with a good deal of hope for thinking about the future. Today, we're going to hear a little bit about BC's strengths as a leader in discovery research. Also, we're going to hear about some of the limitations and obstacles that exist for researchers as they attempt to translate their discoveries into real-world treatments for patients. As a research university, of course, UBC is at the core of the process, something that's called translational medicine. This is a collaborative, multidisciplinary endeavor that requires the involvement of academia, industry, and people right across the healthcare system. It's a process that brings discoveries from what they colloquially describe as bench to bedside. So from the laboratory bench all the way to someone's bedside as they're looking to get help and support. For today's discussion, we're going to be focusing on the earliest stages of the translation process, the steps leading up to clinical trials, but we will not be delving into some of the other checks and balances that happen along the way. We're going to avoid the politics of all of this because you might be surprised to know, or not, that there's a lot of politics involved in some of this as well, but we're going to leave that for another day. I'm going to offer you a few definitions just so that we're all talking about things in a way that everyone can understand. So discovery research, this is known as basic science or fundamental research. It focuses on a generation of new knowledge and the uncovering of underlying mechanisms behind health and illness. This is the work that leads to scientific discoveries and is primarily based at universities. It's where stuff starts to happen. Questions get asked and discoveries get made. Commercialization. You might not be surprised to know that commercialization is understood as the mechanism by which new treatments and drugs are mobilized so that they can reach patients. Companies take on this responsibility. It has financial risk attached to it. For clinical development, for testing products, for obtaining regulatory approval, and then for manufacturing and distributing things at scale. This is where collaboration starts to happen. Universities like UBC start to create spin-off companies and then license intellectual property, and then that grows and becomes the genesis upon which things like new vaccines, you might be familiar with Pfizer, Moderna, and so on, companies that produce vaccines sort of come into partnership with universities and so on. So then clinical applications, and this is key as well for the people that you're about to meet. Clinical applications put commercialized products and treatments into use by health practitioners in the health system so that patients can realize the benefits of the research. So this is the bench to bedside mechanism. So the whole sphere of areas of interest that we're going to be talking about really live in translational medicine. So how do we take these discoveries, the questions that are being asked, the discoveries that are made, and how do we understand what's going on in the lab as a way for us to understand what's going to happen to create a positive and helpful healthcare system for all of you and us listening. So we're talking about the process of translating discoveries that come from basic research, transforming those discoveries, made the lab bench into treatments, drugs, devices, and diagnostics that can then be translated into improvements for health, and they can ultimately save lives. So this is a snapshot here of the long road. And what's really fascinating, as all of you are aware, is how compressed what usually is a long timeline. What has happened over the last year and a half has really surprised many people, as this timeline has been accelerated in so many different ways. This snapshot is the average timeline for translational medicine. And while we don't need to get into the details of this diagram, it can take 15 to 20 years sometimes to move from one stage to another. And it can be excruciating for those people who have a health condition, or a loved one who has one, or a scientist who knows that they're right on the verge of it, having to wait. So it's just an exercise in understanding what's going on here and how quickly some things are moving these days. So for example, despite the dramatic increase in research in Alzheimer's disease over the past couple of decades, more than 200 clinical trials of promising candidate drugs. Until recently, there was no new medications for this disease, and none had emerged since 2003. So ultimately, extended timelines because of roadblocks and time spent navigating systems have sometimes led to challenges with patient health. So we're not talking necessarily about safety measures. They're, of course, essential to the process. We're talking about some other things that happen along the way that slow down the process. So following up on some of this, we really want to have a conversation that invites you to listen into the conversations that are going on within the Faculty of Medicine. So the faculty has a really bold vision to radically accelerate the development of new therapies that can be delivered to clinic in human trials in a more quick and efficient way. With us today, we're lucky to have three experts I've mentioned before that are with the Faculty of Medicine, and they are working in their own locations, and they're going to be sharing with us some of their insights along the way. This slide that you see before you is an invitation to participate. If you go on your telephone or on another browser or on your mobile to slido.com, this is a platform where you can actually tap in questions to us that we'll be able to knit into our conversation. So really want to encourage you to participate. Your passcode is very strict, one word, transform health. So you can tap that in, and suddenly you'll see before you an opportunity to pose a question. And I'm going to be using my own mobile to look for your questions and then bring them forward so that we can share them with the people who are our special guests today. So here's what's going to happen. As you're busy getting your Slido ready, we're going to have one-on-one conversations. They'll be brief with each one of our three special guests. I've got a couple of questions that we'll ask these esteemed experts. They'll talk a little bit about their own thinking about their work, their own discoveries. Maybe they'll even give us some insights into how they feel about some of the work that they've been able to do so far. And then we're going to have a wide-ranging conversation with all of us together and have them get an opportunity to really speak with their colleagues about some of the work that they have done. So I hope that's as clear as mud for all of you. The bottom line is this: the Faculty of Medicine is doing some extraordinary work, and they're hoping to do more of this extraordinary work so that they can help to create new and exciting discoveries and help to support people's health and well-being in a much more efficient way over time. And the three people you're about to meet are all participating in that now and are going to be doing so in the future. So I'm very, very excited to welcome each of them. So let me begin by our first guest, Dr. Pieter Cullis. Pieter is a triple alum from UBC. Many of you have probably seen this man's name a lot in the last 18 months or so. He's been in the news a good deal. Dr. Cullis is a professor in the Department of Biochemistry and Molecular Biology. He is also a member of the Life Sciences Institute at the University of British Columbia. He's also one of the founders of Acuitas Therapeutics, the UBC spin-off company that actually helped to develop the lipid nanoparticle technology that allows the Pfizer and Moderna mRNA vaccines to enter human cells. So yes, this has been a big year for Dr. Cullis. Pieter, in your own words, I want to welcome you, and I want you in your own words to really talk about what this past 18 months has been like for you as somebody who's really been on the cutting edge of these technologies that are literally making their way into our arms as we speak.
Thanks very much, Catherine. Yeah, it has been quite an amazing, last 14 months. But it's actually, as they say, I mean, I've been involved in this field in one way or another now for 40 years. And so part of what's going on is it takes 40 years to become an overnight success. And I'll just go through that very briefly by way of introduction as to how it is we came to this situation where we did play a role in this in the mRNA vaccines, which are clearly having a major effect as far as the pandemic is concerned. But in the early 80s, when I joined the biochemistry department here at UBC, I was first concerned with delivering, well, actually I was working on very basic problems to do with biological membranes. But the first application that we had was trying to deliver cancer drugs more specifically to where they're needed. As you're aware, when people take a cancer drug, I mean, at this point, about 0.001% actually gets to the tumor site. The rest of it goes elsewhere in your body and has very nasty side effects. And so what we were developing, and still are, is to some extent, are systems that can get to the tumor much more specifically. So we got actually 3 drugs approved by the FDA and the European Medical Agency during that time. But in the middle 90s, we decided to change our, to include the possibility of delivering DNA and RNA based drugs, nucleic acid based drugs, which are much, much larger than the usual drugs that we're used to. They're so-called small molecule drugs like aspirin and ibuprofen and those sort of things. And so the reason that we use these small molecules is because they're the only things that can get across cell membranes and affect something that's inside the cell. So really what we had to do was develop a system that could first of all encapsulate these very large molecules, at least as far as other drugs are concerned, and then find ways of getting them to be taken up and get into the interior, what's so-called the cytoplasm of a cell, so they could actually have some biological effect. So it's been a long saga, 25 years. And we borrowed very extensively from our basic research in that process. The properties of lipids and membranes, the ion gradients that are exist across membranes, and some of our experience there really it did inform what we ended up doing. Now, what we started to do was to first of all we found a way to encapsulate nucleic acid-based drugs in lipid nanoparticles. And then we embarked on a collaboration with a company in Boston called Alnylam between about 2005 and 2012. And this was to deliver a nucleic acid-based drug called small interfering RNA to silence what is a pathogenic gene, in other words, to inhibit its production of the protein in the liver. And so that went very well. And the drug went into clinical trials in 2012. And then through another company that I'd co-founded called Acuitas, we started to work on saying, okay, if we can deliver the small interfering RNA, which by any normal terms is a large drug. Maybe we can deliver much larger ones, which messenger RNA is a heck of a lot larger. uh, and the and so we packaged them up in a similar kind of lipid nanoparticle and found, yes, we could, if we injected them intravenously, then they would start producing a protein in the liver.
Let me fast forward you for a moment, because I know you made a number of discoveries along the way. If I can fast forward us to COVID-19, when the pandemic was identified, did you immediately have a light bulb going and thinking our discovery might be able to play a role here?
Well, this is where serendipity comes in, right? And I was so we decided we had these systems that would make a protein in the liver. Well, then we were contacted by an immunologist at the University of Pennsylvania, saying, a guy named Drew Weissman, who said, well, gee, I'd really like to try these as vaccines. And and I turned out to have absolutely spectacular effects for Zika virus and influenza, at least in animal models. And that got us working with actually two German companies, one called CureVac and the other called BioNTech. And the CureVac ran a clinical trial and showed that these systems could produce a very effective vaccine against rabies. That came out about January of 2020, and BioNTech was working with Pfizer on an influenza vaccine. Of course, all efforts changed in the January-February timeframe, saying, okay, we have to get a vaccine for SARS-CoV-2 for COVID-19. And so it ended up that the lipid nanoparticle we've been developing for some of these other applications, cancer vaccines amongst others, was incorporated into the Pfizer-BioNTech product, which went into clinical trials. I guess it was March or April of last year, and then there was a real wow moment. Well, first of all, we realized that the huge effort that was going into this back in March or April of 2020. But then in November, when the results came out and it was 95% effective, and I should say that, a very large proportion of the improvements of the ability of these things to, the mRNA as a vaccine does not work without the lipid nanoparticle. So, right. This is, we played a pretty important role in all of this. Anyway,
so you kind of skipped over the wow moment. And I'm gonna.
I was going to say, well, really, the wow moment was 95% efficacy, and that was announced in November. I mean, when we heard that, we thought, "Good Lord! I mean, this can't be true, right?" So, because the whole world was waiting on that moment, as the initial results from the vaccine trials came in. So it was it was just. Really that was an incredible moment. We had a bit of champagne at that point, and we drank it virtually.
Hopefully more than a bit.
Yeah, now to see that there's 3 billion doses that are going to be put into people's arms this year is beyond belief, right? I mean, and these are compounds. Acuitas is based on the UBC campus, and so we synthesize the molecules that go into the lipid nanoparticle right here at UBC. And so, yeah, we can all take some pride in the fact that it really was UBC science some of the spin-off companies that UBC has encouraged that have made a pivotal role in this. Without this effort, without this expertise, we wouldn't have the mRNA vaccines.
Right. I'm going to pause you there and bring in our other panelists, but I'm really excited to ask you more about this. And it must be disorienting to think about this incredible role that you and your team have been able to play, like unbelievable on some
it
level.
Absolutely is. I mean, just before this webinar, I was talking with Ugur Sahin in Oslo, his wife. And they're the people that founded BioNTech, right? So they're in Germany, and they're in exactly the same space. I mean, your life has changed enormously as a small biotech company, and then all of a sudden you're playing this pivotal role in a global context. So it really is. It really is. Yeah, the other part of it, of course, is it's not just vaccines. That's just the tip of that's the tip of the iceberg. The other applications of mRNA-based therapeutics is it's essentially pretty much any disease you care to name. And so we're busy really pushing that forward, exploiting the possibilities there.
Well, it's been fascinating to watch you and listen to all of the conversation around the research, and I'm very excited to learn more in the coming minutes. So thank you so much for getting our conversation started.
Thanks, Catherine.
You're very welcome. So Peter, of course, is going to remain with us, and he's going to be participating in the conversation that we have going forward. So I'm now going to introduce our second speaker today, Dr. Megan Levings. Dr. Levings is the Canada Research Chair in Engineered Immune Tolerance, professor of the Department of Surgery and the School of Biomedical Engineering at UBC, and the lead of the childhood diseases theme at BC Children's Hospital Research Institute. She studies immune cells called T regulatory cells and their potential applications as next generation cellular therapies. She's internationally recognized in the field of human immunology, and her work has incredible potential implications for transplant rejection, for autoimmunity, for chronic immune medicated diseases such as diabetes. And she's just got her fingers in so many pies. And again, we're just going to scratch the surface with Megan, just to talk a little bit about her work. So thank you so much for saying yes to us, Megan. Great to have you with us.
Thank you very much.
When you listen to colleagues like Peter talk about 40 years of work and having a discovery that really is groundbreaking, life-changing, what do you as a young scholar scientist think about?
Yeah, well, it's humbling and inspirational at the same time. You know, on the one hand, I think, wow, the discoveries coming out of Pieter Cullis's lab are amazing. There's no way I could ever do anything like that. But at the same time, it also opens your imagination to think, you know, I guess I'm 20-ish years, maybe a little bit more behind Peter in my career. You know, maybe when I'm at that stage, I will have brought one of my discoveries to that point and there will be impact. So it's pretty exciting, actually, to hear about what's possible when you're in the academic context of the university, surrounded by so many amazing young trainees and other people to work with.
Let's talk a little bit about your work, specifically immune-based diseases, and what can happen in terms of your work and how that translates into not only discoveries, but also into the way that people receive care and services. Can you, in a brief way, describe the work that you're doing?
Sure. So I'm an immunologist. So I study white blood cells, and a very special type of white blood cell, as Catherine says, called a regulatory T cell. And their job basically is to turn off immune responses. I think most people don't realize that the immune system actually spends most of its time trying to keep things quiet and not being active. You know, on a daily basis, we're not trying to fight off some horrible pathogen or anything like that. We just want to be quiet, and we don't want to respond to all of the proteins our own body is producing, or all the food we're eating, or all the things we're breathing into ourselves. And so the immune system, and in particular these cells I work on, regulatory T cells, are continually keeping things quiet. When a virus comes along, the system overwhelms the function of the regulatory T cells. They're still there, but they stop being able to keep things quiet. But on a day-to-day basis, these cells are preventing autoimmunity, preventing allergy. If you had a transplant, they hopefully would be preventing graft rejection. And so our idea is that if we could harness the natural properties of these cells, either by finding ways to boost their natural function inside the body, or what my lab is particularly interested in is taking them out of the body, expanding them in a laboratory setting, maybe genetically modifying them to give them particular powers that we want, and then giving them back as a cell-based therapy. And that way we could harness this natural function of the immune system to reprogram inappropriate immune responses like an autoimmunity or transplantation rejection.
What have you discovered so far with the idea that you might be able to manipulate these cells to behave in particular ways depending on the particular circumstance? What have you discovered so far that gives you an idea of what could be possible?
Yeah, so I think probably many of the participants now have a bit better understanding about immunology after the whole COVID thing over the last year. And you've probably heard a lot about antibodies and maybe even the T cell response. And so the immune system works by having each cell in your body has a particular receptor. It's like a lock and key mechanism where you are expanding particular populations of cells that have a specificity for a particular, usually protein. And so most of the early studies of the cells that I work on, regulatory T cells, were done with cells that weren't selected for any particular key. It was just, whatever cells came out, people were putting them back in. And that means that you weren't really harnessing the whole advantage of the immune system, which is this lock and key and memory system. So what we started working on actually about 10 years ago now was how you could genetically modify a regulatory T cell to redirect its specificity towards a particular protein that you wanted it to respond to. It's a concept that had really been pioneered in the context of cancer immunology. Some of you may have heard of CAR T cell therapy, which has really transformed aspects of treatments for cancer. And so we took that concept and say, hey, can we use CARs and also put them in regulatory T cells, but for the opposite purpose, to specifically shut off immune responses rather than turn them on. And I think the moment of, you asked Peter about the aha moment. Yeah. The moment when I knew this was going to work was when a student in my lab, Rami Hopley, was doing a PhD along with an undergraduate trainee, Vivian Fung, did this experiment where they, in an animal model, had done a transplant, and we had labeled our genetically modified Tregs with a color that we could see when we then put the animal under an imaging machine. And we saw that 100% of our genetically modified cells went to the transplant and didn't go anywhere else. And it was this incredible image that was the aha moment when I knew it was going to work.
So did you turn the screen off and turn it back on to make sure you weren't imagining things? Or how did you center yourself to realize, oh my goodness, we might have found something?
Yeah, well, it was in a lab meeting. So everyone was just, it was just incredible when we saw that. And it was so clear. It was, I think maybe the general public doesn't realize that a lot of science is things that don't work, experiments that aren't clear, not a black and white conclusion. And then every once in a while, you'll get this experiment that's just, this was what we thought should happen, and it happens, and it is so clear. It's just one of those moments.
Wow. So you've got this laboratory moment, you've got your crew of people realizing the possibilities here. Do you immediately start thinking about, okay, how can we pull it out? How can we find a partner? How can we work collaboratively to develop this so that it gets out and starts to grow in the world? Like, walk me through just for a moment or two, the process. Like, how do you go from that moment to what are we going to do about this?
Yeah, well, I have to say my answer to that was no. So there was this incredible discovery. And I guess, mostly at that moment in time, which was like 5 or so years ago, I was totally focused on getting my students through their PhD degrees, getting our papers published. And I hadn't really thought that much about a potential, when we write grants, you talk about, and one day we'll translate our whatever discovery into clinical trials, but you never really, at least I never really believed that I would do that. And so we went on our way and we published this paper. And then literally the day after, I started getting phone calls from people who were interested in it. And in particular, one small biotech company that was very interested in it. And that's when my journey along this translational pathway started.
Well, I'm so fascinated to hear a little bit more about this, and also to hear from our other panelists, just sort of about how they move from laboratory into the community, into the sort of more commercial, back to the public, and so on. So lots more to learn and think about. Did you imagine, as a person starting to study immunology, that you'd be on the cutting edge of discoveries like the ones that you've participated in so far?
Definitely not. No, I mean, I guess well, yeah, I don't think anyone really imagines they'll reach that point. And also, like Pieter Cullis said, it's like one moment that reflects 40 years of previous work. And so it's not, you don't really start off being fully confident of where you're going to end up. It's years of work that get you there. But it's pretty exciting now to be at that moment where you realize that you do have a possibility to take something forward that's going to make a difference.
Wow. Well, I can't wait to listen to more. Thank you so much, Megan. I'm going to bring in our third panelist now as we continue this conversation. This university is just full of brilliant humans who have such good questions. And I sometimes think we spend so much time talking about the answers that we forget that there's beauty in asking questions over time, whether it's a 40-year career or 20 years so far, or the career we're just about to find out about now. So a very big pleasure for me now to welcome Sriram Subramaniam to this conversation. He is the Gobran Khorana Canada Excellence Research Chair in Precision Cancer Drug Design. He was recruited to UBC just a couple of years ago, 2018, from the U.S. National Institutes of Health in Bethesda, Maryland, where he was a senior investigator and founder and director of the Center for Molecular Microscopy and the National these ones are the hardest. Cryo-M facility, pardon me, at the U.S. National Cancer Institute. I should make you do this, Sri. His research is incredible. It involves cryo-M methods and cryogenic electron microscopy as related to drug design. Now, this is a technique that allows imaging of proteins at a near atomic resolution, so teensy, weensy, 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 after first cooling them to a cryogenic temperature, and then they play around with it and manipulate it and figure out ways to mobilize it to create pathways for healthcare. So Dr. Subramanian is the first to demonstrate the use of cryo-M to obtain protein and drug structures at resolutions relevant for application to precision medicine. His research team used this technique recently to obtain structural images for COVID variant that allowed scientists to better predict and prepare for its impact. And his team is also actively working to describe structures of spike proteins from current variants of concern. So we are talking to three extraordinary human beings today. So I'm so excited to welcome you, Sriram, to this conversation, and to UBC, because we've never met before today.
Very nice to meet you. Thank you very much, Catherine.
Thank you. So we're talking about the excitement of this science and you know, all of us living through this kind of ridiculous circumstance of a global pandemic. Situate your own knowledge for us, if you will. Where are you right now with your work and discoveries, and how does that work within the context of the investigations underway with regard to COVID?
Yeah, thank you. That's quite a lot to unpack across many time zones and disciplines. Maybe just a little bit of background to the technology itself. Cryo-EM is something that's become relatively popular recently, but I think perhaps nothing has illustrated the power of imaging at these kinds of resolutions where you can visualize molecules and atoms as COVID-19 has, because of the visibility of the application. The pictures that we've all seen on news media, news coverages, they all show these multicolored pictures of spike proteins sitting on viruses. Of course, in reality, they're not colored. They're just atoms sitting there, and we just highlight them just for visual effects. But the history of this, the ability to visualize these molecules goes back many decades. You know, Peter spoke about 40 years, and Megan about 20 years. You know, I think in some of the foundations for the ability to image proteins, viruses, cells, at these relatively high resolutions goes back several decades, with methods developed primarily in Cambridge by Richard Henderson, Nigel Unwin, and their colleagues, where they showed that you could actually use electrons as a mode of imaging. People at that time knew that you could use X-rays as a source of imaging protein crystals. And we all know how you can use light microscopy to look at cells and things in real life. But the idea that you could use electrons to image proteins was a concept that really was established in the mid-70s. And it took several decades in the 90s before there was even the prospect that something useful might come of it. And when I came to the NIH, which was about 2 decades ago, I had really done very little work in this. This is not something that I actually had worked on. I used to work on fruit flies. But that's what I had started my career on at Johns Hopkins University School of Medicine. But I was taken very much by the possibility that this could be useful. And I was hired at the NIH solely on promise that maybe this is something that, something may come of this one day. And they took on faith that, they asked me, had I actually done any of this before? I said, no, but I thought there was something there. And it was strictly on the potential that something may come of it. And of course, at that time, 20 years ago, the word cryo-EM, which I was pleased to see you had difficulty saying, and that's not my name that you had difficulty with, but it was not really a word that people used because not enough people believed that much would come of it. I think our wildest hope was that maybe we would get somewhere to visualize proteins and so on. But the fact that a few years ago, we could get to show visualize proteins at near atomic resolutions was just unbelievably exciting. The fact that we could get there, the fact that we could see water molecules in these proteins. But then comes COVID-19, and all of a sudden, across the world, dozens of laboratories actually are using these methods to look at the spike protein, to look at the virus as a whole. And more recently, in many labs, including our own at UBC, we are trying to understand these mutations as they evolve and understand why is it that these variants of concern are spreading so rapidly. What makes them more transmissible? Why did the UK variant suddenly take over? And now the Delta variant, for example, why is it that it's suddenly so prevalent? So the answers to these riddles lie, at least in part, in the structure of the virus and the structure of the virus and the proteins that it displays and the ways that it learns to evade the immune system. These are really fascinating struggles, if you wish, that are taking place in evolution, the virus trying to get in, the immune system trying to fight back. And, our hope is that we can make a contribution to understanding the origins of exactly what it is that improves the virus to be fitter and to be more transmissible, and as a consequence, understand how can we fight it better. So a lot of basic science, but with very much with the application in mind.
You are at the top of your game, obviously, in doing the work that you're doing. You chose to leave Bethesda, Maryland, a very well-funded and politically highly regarded laboratory, and come to the University of British Columbia. Why?
The NIH is a great place. The science was fantastic. There were great colleagues. There's about 1,500 labs of people doing enormously interesting things. But I was really driven by an interest in translation. In some respects, and I would speak about this to others, we were publishing good papers and good journals. And, as scientists, very often we look for publishing in journals with, high impact factor. But I was deeply driven by translating this into real impact. So the transition from having an impact factor of publishing journals with high impact factor to actually having societal impact, that was very much at the core of my own interest in moving out of the NIH, out of the government. But UBC, I think there was a hunger here, and it's you hear Peter and Megan, you see it throughout. There's a tremendous hunger to make an impact, and it was palpable. I mean, it was not difficult at all, and I'm extremely happy to be here. I'm glad that they actually made me an offer to begin with.
Yeah. Well, maybe this is a good moment for us to invite the panelists back, and we'll have everyone on screen. You're welcome, those of you watching, to participate on Slido and to pop your questions in. I'll keep track of them as we go. and just join in this conversation that we're having with three of the leading scientists, three of the many leading scientists who work with us on the University of British Columbia campuses to ask questions, make discoveries, and then share them with the world. And their goal, as you've heard from each one of our speakers today, is to make change for people in the world and translate their knowledge into action and change for people who have diseases or who are subject to something like COVID-19. So Slido.com is the platform. So on the World Wide Web, www, if my mom is there watching, as she often is for these things, www.slido.com, Mom, and then you just type in the passcode Transform Health, all one word, and you'll be able to ask us your questions and we'll bring them to our panelists. So maybe we'll ask everyone to reappear back on screen. And I'm curious if the three of you have had an opportunity to meet each other, as the kids would say, IRL, in real life.
That's a new one.
In the last year and a half, or whether you, like everyone else, has sort of been keeping to yourself. So this might be a great opportunity for you to say hi to each other.
So I think just before the pandemic started, we had invited Sriram out to give a talk. My lab is at the BC Children's Hospital. So I was fortunate to meet him. What was the acronym? IRL?
In real life.
Just in real life, yes. But since then, it's been a lot of Zoom, and I've never had the opportunity to meet Pieter Cullis in person, but I'm really looking forward to that one day.
Actually,
I've got to say that there's possibility now for additional collaboration. After having heard what Sriram and Megan have had to offer here, I think there's. You want to do in vivo CAR T-cell therapy, Megan? I think we have some ways of doing that.
Okay, that would be cool. Yes, and I want to use your lipid nanoparticles to do gene delivery for our Tregs as well.
Yeah.
Okay, now we've got to keep you three away from the jargon, because it's not only difficult to pronounce, it's really hard to figure out as a non-medical person. So thinking about creating a space that is welcoming of ideas and discoveries. And I love the way that you described it, Sriram, as riddles, solving riddles along the way. That's kind of the language that Michael Smith used to use, the wonderful man who the Michael Smith Laboratory is named after at UBC. He would talk in these great expansive descriptions about making puzzles fit together and thinking about science as riddles. So as the three of you were listening to each other, what comes to mind in terms of the questions that you have about each other's research and the ways in which you see that knowledge being translated and made available to the general public? Maybe I'll start with you, Pieter.
Well, first of all, as Sriram was, cryo-TEM is a technique we use very routinely, and I think that we need to chat a bit about we use this to examine the structure of these lipid nanoparticles. And it's a mainstay technique for us to really see what we've got. It's absolutely fantastic. As you mentioned, the progress in that field has really enabled a tremendous number of things, and the lipid nanoparticle field is really deeply indebted there. Sorry, what was your question again, Catherine?
Just wondering where these possibilities live for you as scientists, thinking about what other people are doing and thinking about the ways in which your discoveries can be then translated and have an impact in people's lives.
Yeah, well, with the mRNA therapeutics, it's really one of the problems there is to focus, as the opportunities are so enormous. The cancer vaccines are a possibility as the vaccine area, HIV and rabies and Zika and all of those, influenza. We're going to see vaccines coming in all those areas in addition to the COVID-19 pandemic. But the other therapeutics that are possible are just incredible. Mainly because imagine a child being born with a enzyme deficiency. This is, these are conditions that happen all the time. Currently, the only way, the only way we can correct that is using a virus to produce that protein following, say, an IV injection. The LNP, the lipid nanoparticle approach allows you to, the virus approach is quite complicated. It costs you $1 million and takes a year, which is often way too long if the child is in need of something that happens much more immediately. But with the mRNA technology, you can use the liver as a bioreactor. And so just put mRNA into a lipid nanoparticle, inject that, and this can be produced in a matter of a couple of months. And then you can envisage the, that protein being produced by the liver and the child being, basically surviving. So these are the sorts of immediate things for rare diseases in children. And but it goes way more than that. I mean, there's everything from Alzheimer's. The brain possibilities are huge. As are, we're now getting systems that can go to bone marrow, so leukemias and sickle cell anemia and all of those things become logical targets. It really is a spectacular. The amount of investment that's going on in this area globally is unbelievable. There's companies now with market capitalizations approaching $100 billion in this using lipid nanoparticles for one application or another. So it's taking off globally, and one of my ambitions is to make sure that some of that stays in Vancouver, so that we really have a robust, industry, the therapeutic industry going on here.
Okay. The questions are starting to pile into Slido now, so I'll turn to those, but I really want to welcome each of you. If you have questions that you want to raise with each other, or you want to comment on something that someone else has said, please feel really welcome to do that. So this one comes from an anonymous typer. What are the challenges faced by BC researchers pursuing commercialization? Do the challenges differ by career stage or field of research? Maybe Sri, maybe you would touch on this one first as somebody who's sort of new to this environment.
Thank you. It's a question I think that many of us think about in terms of, especially in the context of translation, exactly where the challenges are, because in a scientific context, often you think of where the gaps are. But when it comes to translation, you focus more on, okay, what are the challenges to go, to bring this forward? And for me, I think I think there's already one thing that all of us at UBC have in our favor, which is a very supportive ecosystem that favors faculty members and staff moving forward translationally to being entrepreneurial. So I think that's a big plus already. But in terms of asking where the challenges are, I'm actually particularly honored to have the Govind Khurana chair, and he was my mentor, postdoctoral mentor at MIT, and was a very significant influence in my own academic career. And the very first day, I remember showing up in his lab, the bench that was assigned to me had a little thing that said, "The difficult we do today, the impossible tomorrow." And if you look at his, his career, I think he didn't worry about things that were deemed to be impossible. He just said, well, that's for tomorrow, just do the difficult things today. So I think in that spirit, I think there's a lot of opportunity. I think we just have to just do it.
Wow, what a beautiful quote to leave on your chair. Wow. Megan, that's a little bit, it reminds me of what you said earlier, just not being able to imagine the thing to come. But if you keep open to the fact that things are possible. One of the next questions comes from someone saying, what's needed to accelerate these sorts of discoveries? So we've heard note about open-mindedness, collaboration, this sort of sense of discovery and wonder. What else do you think is needed to ensure that these discoveries keep happening?
Yeah, so I guess one of the big challenges in my field, the field of cell therapy, is there's just the huge expense and logistical complexity of manufacturing cells in a lab, and it requires very highly specialized infrastructure and then particular infrastructure in the hospital to deliver this product. And when I embarked on this journey, none of that was available in Vancouver. In fact, it still isn't. And so that meant that we ended up partnering with external people to develop the technology, which was fantastic because it moved forward very quickly and is being, is going to be tested this year. But it is somewhat disappointing that I wasn't able to do it here at UBC. And so something that I'm, Sriram said, you're doing the hard thing now, tomorrow you're going to do the impossible. So I spent a few years just being frustrated by not being able to do it here. And finally I'm like, okay, just gonna have to work on, getting the cell therapy facility here in Vancouver and getting the funding to do that and getting what we need in place. It's gonna be a lot of work, but I think the thing that really kind of pushed me over the edge is the new School of Biomedical Engineering and all the new colleagues that I have now that are also working in this area. So it's not just me by myself anymore, it's a team of people. And we have a critical mass to really take this forward. So it's exciting.
Peter, do you need a bit of a stubbornness when you're doing this sort of work? When you think about the fact that you can only do so much in the laboratory, you actually do need these other steps along the way to be able to scale your discoveries and turn them into something that can become an action.
Yeah, I mean, I guess you need to be willing to step well outside your comfort zone, and that's which is a difficult thing to do. But I was, I graduated, my PhD is in physics, and I decided I didn't want to do physics anymore. And so I went to, my postdoc was in biochemistry, so I went to this biochemistry lab, and I didn't even know what a protein was. I had DNA, whatever. I mean, I was completely naive. I knew a bit about NMR, and so therefore I had a technique that I could apply to some biological problems. And after a year or two, I actually found I could hold my own with people that were talking in more biological terms. And so that was I found that very reassuring. Of course, then it led to, okay, well, gee, I survived this environment, maybe I can start a company. And so that was, again, moving outside my comfort zone. I've never taken the course in biochemistry, and I certainly have never taken the course in business. But you sort of have to go and do it. And then you learn. I was going to say, you can learn the hard way, and but you certainly learn more quickly if you're actually immersed in something where you really know that you're out of your depth, that you don't know what you're talking about. So it's that's what I did try to encourage in people, is that just step well outside your comfort zone, then you bring things to other disciplines that are not that they often don't have. So you can there's a new perspective you can bring to that. That new area that you're engaged in. So that's what I really encourage.
Right. Sriram, I was watching you chuckle as Peter was speaking just there. What were you thinking about as he was talking?
I was just thinking, just taking Peter as an example, what he probably doesn't know is that in my PhD work, I was reading the papers he published on hexagonal phase transitions, Peter. I worked with Harvard McConnell. My thesis was on phase transitions, and with Megan, we actually are co-investigators on a Wellcome Trust grant. Although we haven't met in this context, Peter Zanstra is involved. We're doing things. So I think the length, over time, you have connections with people that they're like Megan said, they're these T-Rex, they're sitting dormant. They suddenly come alive, in context. So the vehicle phase transition suddenly, comes alive, or, the connections that you build up over time, they're all rich and unique and really very, very powerful. And I think those of us who've been in science for some time, I mean, that's probably the most enduring thing, which is one is, of course, stepping outside your comfort zone. But each time you do it, you bring along knowledge from another field. And, if you're willing to fall flat on your face a few times, and learn to get up, and, eventually, it works out. But it's the willingness to take those risks that I think is what keeps us going. I certainly feel. I mean, there's some people that know so many things, but unless you ask and you step outside, you won't find out how little you actually know.
Well, it is it is a bit intimidating to read your CVs, the three of you. You know, when Ophelia, one of the organizers, sent the briefing notes, I'm thinking, oh my, these people just do everything. But it spoke to, I was thinking about, well, as a scientist, you could pick a thing and do that thing for a long period of time. But all three of you have picked a bunch of things, and your curiosity keeps taking you new places. So Megan, I'm going to put this one to you. How do you not become complacent with the thing that you're working on today? How do you stay fresh and open to the possibility of the thing that might come tomorrow?
So yeah, I guess you're right that I think you introduced me as having a lot of things going on, which is very accurate, because I mean, I guess when you're a scientist, you just are intrigued by new problems that come your way. And these regulatory T cells I work on, I guess what is amazing about them is that they're involved in everything. So initially, we understood them as just being a very immune-centric thing, regulating other immune cells, but now they can regulate cardiovascular disease, they can regulate Parkinson's disease, they can regulate Huntington's disease. It's incredible. There's a paper that just came out showing that their function is essential for recovery from stroke. And so when it's so hard to not keep excited and keep moving forward, even if it takes you in different directions, because you can just realize all these different possible applications of the things that you're working on. Right. But you do need to have some focus, because you can't be an expert on 100 different diseases. So I definitely go through phases where, okay, now we're working on too many things. Let's rein this back in, focus. And then a little bit later, I'll be out again and then need to focus. So it's always finding that balance.
There's some appetite on Slido to to have each of you talk about the very specific things that you're working on and the possible implications of those discoveries. So I want to get to that, but there's a broad question that I've been wanting to pose to you, which is also shown up in the chat. So I'm going to pose it now. How do you think the world's recent recognition of science and scientific research from COVID-19 will impact the field of scientific research in the future? Because I do think, even as people post those ridiculous little selfies with their Band-Aid and their COVID-19 stickers, they're saying, thank you, science. We haven't seen this before. So what do you think the possibilities might be as a result of people recognizing the importance of scientific research in the world? Peter, then Sri, then I'll ask you that as well, Megan.
Well, I'm quite hopeful, actually. I do think this is going to make a big difference, and not just in Canada, but worldwide. If you see what they're doing in the UK, I mean, the emphasis on science as part of their recovery is huge. Really an enormous investment, and there's rumblings of the same thing going on in the European Union. Haven't seen that quite to the same extent in the States, but the investment in from a company point of view in the life sciences is stupendous. So yeah, I think this is. There's a couple of things. I mean, one is the impact it's having, but also the very rapid response. You know, the pandemic hits last January, February, and then by November we have a vaccine. That is absolutely spectacular, and you know, so people now can see that. You know, you were pointing out at the beginning, Catherine, the 15-year potential time frame for a new drug to evolve. I think we're going to see much more of this. That you know, for newer drugs, particularly the mRNA, the nucleic acid-based drugs, a much faster translation, and that's going to bring it. You know, the notion of more personalized therapies, and of course that in turn is going to mean that you know, individuals are going to be impacted more and more by these kind of medicines, which I think in the end is going to be reflected by, you know, increased funding when people actually see the benefit of these new medicines.
What's your thinking on this?
You know, I think it's broader than just the science. I would say the science and the technology are intimately connected to each other. So, you know, if I look at the last decade, certainly you could say this in previous decades, but the pace of science and technology that continues to accelerate. So if you look at the last decade and the coming decades, I think technology should be viewed really not necessarily as a, as an instrument or hardware or software, but as a strategic asset. I think that is the key. If you look ahead, and I think UBC, certainly Faculty of Medicine, I think appreciates this very deeply, that the technology goes hand in hand with science, because as these technologies reach a certain level of performance, they accelerate and enable scientific discovery, which in turn drives the development of these technologies. So I think the recognition that these scientific advances have such a transformative impact on society is one thing, but there's something below it that is not passive. It is not a pre-existing technology. These technologies are evolving constantly, but they're driven because their advances are driven simply by virtue of the fact that they're actually having an impact. The science is having an impact. So I think that's where I see certainly UBC very well poised for the future to recognize that if you understand the interplay between both those and recognize that each of them drives advances in the other, then I think it would make for a very vibrant sort of contribution of these kinds of areas to society going forward.
Right. Thank you. Okay. Megan, your thoughts on this one, this recognition of science, and then Sriram points out technology as well, and the way that that people might have a newfound appreciation and how that might translate into change.
Yeah, I think the general public definitely has appreciated more the pathway and the importance of the fundamental academic research. And you said you didn't want to bring politics into it, but with this question, it's impossible to not mention that everything we do and everything we discovered was from government-funded research, by and large, the initial observations. And so I hope that the politicians come true to their promise to spend more focus on funding science properly in Canada, because proportionally compared to other countries, we're lagging behind. Canada has this reputation for doing more with less, but we really shouldn't be forced to try and do more with less. We need to have the projects properly funded so that we can have the impact that we want to have. So, but I think it's definitely a step in the right direction, and it's going to be really exciting to see how the academics funding for academic science will hopefully expand, and then the commercialization aspect of that will just be a natural follow-up, because the more basic fundamental science you do, the more opportunities for commercialization and translation.
Right. So there's some questions appearing in Slido around how to make sure that this continues to happen, that the funding goes on. What advice might you offer to a young one considering science? So those are questions that we should probably touch on. We've got another 20 minutes or so. So if people want to use Slido, so www.slido.com, and then you can just write in transform health, write in all one word, and there you will be able to access with the passcode and be able to tap in your questions to us. We're really interested in hearing from you, and also interested in hearing more specific details about each one of our panelists' science. So Sriram, when you wake up in the morning right now, what are the things that preoccupy you? And what are the things that you're putting off for a rainy day?
I have to say that I think constantly about how can we accelerate drug discovery? How do we get there quicker? Why does it take such a long time? COVID-19 taught us something that, in the corona version of something that anybody would have said is impossible. From the fact that there's a vaccine literally on this time scale. I mean, nobody, Peter could not have predicted this. I bet, right? It's not outside the realm of possibility. But with drug discovery, with cancer drugs, I think why should it take so long, and why are they not working so well, and why can't we do better? I mean, I think, and I don't think there's not a single answer, but every little bit that we can do, both scientifically and technologically, I think will help. But you never know. It's millions of hands working together. At some point, somewhere, there will be breakthroughs. But I think we have to work tirelessly to push towards that future that we shouldn't accept these kinds of timescales for the length of time that it takes to discover drugs or their effectiveness.
Do you have a sort of ranking of the projects that you work on a day-to-day basis? How do you sort of keep track of the ways in which your lab is working and moving through your discoveries?
I wish it was I wish that it was so neatly organized and packaged as you say, but it's not. The fact of the matter is, before coming to the NIA, before coming to UBC, for nearly a decade, I had worked on HIV. We did structural studies on HIV. We published the very first structures of these trimeric spike proteins on infectious viruses, and then an influenza, and then Ebola. And that's the phase of work I had stopped. I was totally focused on cancer drug design when I came here. And then in March, suddenly with this COVID-19, I had never worked on coronaviruses before. And the only way we could come to work, myself and as a team in the lab, was to work on COVID-19. So the next day we started to work on COVID-19. That's how and so we didn't really stop working because we used everything we knew and we switched to working on COVID-19. And now we set up from scratch a fantastic team of people in the lab, students, postdocs. The instruments were installed during COVID-19. There was nothing, we were building the facility for these microscopes during COVID-19. Instruments installed, we did structures, and now we've looked at virtually structures of almost all the variants of concern, including those that are yet to emerge, we think. So I think this ability to pivot has been important for us. So I wish it was as structured as you say, that tomorrow we do this, the next week that, but we are pretty much flying, we're doing what needs to be done. Hopefully we're doing the things that are important and interesting for us to work on as well.
Right. I'm curious to hear from each of you too, Peter and Megan. How do you prioritize and adjust given what's going on in the world and given what you're curious about in your own laboratories?
That's a difficult question. As Sriram says, right now we're beset with this surfeit of opportunity, right? There's a whole realm of things that can be done. Now, I do have the luxury of working with a couple of companies, and so there, and I have some influence there. So we can do some work that's more on the translational side, obviously, in that environment. So I have some really excellent people. I should say one thing that is we haven't mentioned, and but that's pivotal for me is long-term relationships in whatever endeavor. I'm still working with people that were in my lab 40 years ago. So in the mid-90s, I was trying to figure out how can I keep this. We had a very good lab with really excellent people. How can I keep this together? And it was very difficult to try and do that in a university environment where everybody, in a sense, has to be separate and developing their own career. And so we started a company in the middle 80s, and a large number, four really pivotal people were working in that company, and then that grew into other endeavors. And the bottom line is we wouldn't have got anywhere near to where we are without that very, very long-term collaborative thing between people that have been working extraordinarily closely with each other. And it allowed us to build up a center of expertise that's unparalleled anywhere else. And so that's one thing I really do encourage, the extent to which one can have very long-term and close collaborations with people that, that's where you're working well and they all have the similar ambition of translation. That's worked well for me.
It's interesting because it's often framed as a race for the answer, or a race for the discovery, or so on. But what we've certainly seen in the last year is how much open sharing of knowledge and information, not to say that there isn't competition, because of course there is, but being able to collaborate in these interdisciplinary and different ways has really helped the process along. Megan, curious to have your thoughts on this one as well.
Yeah, so I guess the initial question is how do you prioritize and what are you thinking about? So just from that perspective, I'm thinking all the time about trying to understand the biology of the cells that we're working on more, I think more passionately than ever before, with the knowledge that somebody is going to be receiving them as a therapy soon, and we really need to know how they work. And so there's just that real push in the lab to do the experiments and get the answers as we translate these findings. So that's very motivational. I'm sorry, my cat now has decided to come and meow in the background. So there's a bit of background noise. And also the ability to pivot. Sriram mentioned starting to work on COVID-19, and in my spare time, I run the Canadian Autoimmunity Sanitization Corps, which is a group of investigators across Canada that's not focused so much on science, but more the methodological approaches to ensure that the science that gets done is the most rigorous and reproducible as possible. And so when COVID-19 turned up, we pivoted and started working on collecting samples from people with COVID-19 and running assays and creating biobanks and working with a network of people across the country. So that's also been exciting to, you know, as both Peter and Sriram said, a lot of the excitement of science is meeting new people, working on new things, understanding how you can take your expertise and apply it in this new context. And it's always inspirational.
Right. Okay, so I've got a couple of other questions here. That it's interesting to sort of think about it as inspirational, because a lot of the questions that are coming through are saying to people, how do you inspire children to go into this? What would you say to a young one who might be expressing an interest in science or research or biotech? Sriram, what would you say to someone who's thinking about this might be a path for them in the future?
You know, I've generally not been too prescriptive about what people should and should not do. It's really difficult to guess. But the one thing that has been true, though, is I think if institutions like UBC in this instance can provide environments that let people enjoy what they're doing. I mean, I think you don't have to be too prescriptive. You don't have to say, well, you have to be in academia or industry or this or that. But if you can stay on the axis of learning and doing things that are important and feel that in some small way you can be part of it, I mean, what more is there? I mean, if that's it, right? I mean, if you could be part of something that has value, that you think you know, doesn't matter however small that contribution is. So I think in that sense, sometimes every once in a while people will say, what's the right path to follow? Should they do this or that? But I don't have any answers, but I couldn't tell you what I might do myself, a year from now. I think as long as I think the goal is to, if you can find places, like I've been fortunate to find places like UBC that let you continue to grow, I think whatever that answer is has got to be the right one, because if you're growing, then it's got to be the right place.
Right. It's interesting, hey, that sometimes people see it as an either or. They think if I choose this path, then I have to stay on it. They don't sort of deviate from that. But you're saying remain open, find a place where you're curious and your heart is filling up. You're experiencing some joy in this, and that will lead to its own benefits and discoveries. What an interesting way to think about it.
Yeah, I think Peter would probably say something similar. That I mean, he probably couldn't have predicted he'd be, working with all these companies. I'm sure he's just doing what he finds entertaining and is fun. I mean, in some respect.
Absolutely. I mean, there's this old statement that if you find something you enjoy doing, then you never have to go to work. It's this, and I think we're really in that boat of this. You get it, the area that we're in, I mean, highly motivating. It's lots of fun. You're working with collaborators here and around the world. What can be more fun than that? It's I think Sriram's absolutely right. I mean, to find something you're passionate about and enjoy and go with it. That's what it doesn't get much better than that.
There is a question that's just appeared about a translational funding gap between research discoveries and company formation. This person refers to it as the valley of death. So just wondering how the three of you contend with that. How do you go out with your discovery and then build a bridge to some funding, to some support, to another laboratory that you can collaborate with? What is that process looking like, and how much support does the university offer you in building those bridges? I'm not sure who might want to take that one first.
Maybe I can start. So, I mean, that definitely goes back to my comment about the Canadian system having not enough funding to really support translational research, certainly not in the way that the NIH supports it. And so just as an example, the cellular therapies that I want to develop, a first in human clinical trial of maybe 20 patients is going to cost around $3 million. And it's not possible to get that amount of funding from CIHR, essentially, which is the big Canadian federal agency that funds this sort of thing. And so you have to be, right now, we have to be really creative and I guess creating intellectual property and finding ways to commercialize, and maybe partnering with a company to do the work rather than doing it on your own, or finding some unique funding mechanisms, maybe outside Canada, to support the work is the pathway that we're embarking on. And hopefully, as we talked about before, the recognition of the importance of science will lead to an increase in funding for these sorts of activities.
Is there a blessing and a curse effect to UBC having had so much attention over the last few years? Now the pressure's on, everybody's watching what all of you are doing and are going to be expecting more and more and more, given the discoveries that have been made in recent years. Peter, is there a bit of a tension at all in terms of the fact that the world now is watching in a different way the kind of work that's being done at the university?
Yeah, this has really been building over quite a few years now. I mean, it's more in the life sciences in particular, as companies kind of starting off with Genentech, but there's now thousands, and so the whole prospect for university investigators to set up. I think Megan's absolutely right. This is one of the reasons why we started up companies early on. It's not just 5 million for a clinical trial; it can be much more than that. If you really want to translate something that you've got an animal model in your laboratory and it's looking pretty good, it's impossible, and actually it's really pretty much impossible in most jurisdictions to raise the kind of capital that you need to move something into a clinical trial and through a clinical trial using money from academic sources. And so that's where I made the decision that, okay, we have to start up a company to if you have any prospects of doing this, and then you can access. I always liken it to the gross national product. If you look at the funding for grant supported whatever, it's about 1% of the GMP. So in other words, there's 99% of the money is out there. You just have to go and make a play for it. And that's that, which is really the commercialization step, and it's certainly getting way outside your comfort zone. I could go chapter and verse on that one, but the university, I find, UBC is very supportive in those endeavors, and I've had situations where we more or less we've nurtured a company in the lab until it gets to the point where it actually might have a product, and then moving it out at that stage. And there are translational monies that you can get for some of that work in your laboratory. But in the end, if you want to really commercialize a drug in the sense of taking it through phase 1, phase 2, phase 3 trials, you can't avoid that commercialization step.
Yeah, I just want to echo what Peter said, which is, if you look in the US at some of the places where, classically would be recognized that being entrepreneurial is almost expected from faculty and staff, MIT, Harvard, Stanford, there's suddenly, that kind of culture. I think I can see that happen at UBC, because at the end of the day, just like science, these kinds of things are done by individuals. And once there's a critical mass of people that think along these lines, I think it will be self-propelling going forward. So I'm very bullish on the fact that this thing can happen here, and that the university has the right mindset and the cultural mindset to enable this, and it could be like a switch, literally, right? You could, it could be, it may be hard. It's certainly hard. I couldn't agree more with as Megan said, it's hard, but we just have to keep pushing, and all of a sudden, maybe this will be, place that these kinds of things happen, and we'll be known for it. So I'm very optimistic that these kinds of things can happen here.
The top rated question in Slido right now is, what is the role for UBC and for education in ensuring medical discoveries make it quickly to patients at speed? So if the university set it as a priority and sort of had that support, as you suggest, that might be part of the answer. Is there a conversation that needs to be had about that, or is it sort of an ethos change? What do you think would be necessary to nudge that along, Sriram?
No, I think it's not a top-down conversation at all. I think it's from ground up. I think it has to come from enough people that have a sufficiently high level of interest. And I think for institutions like this, it's never, I shouldn't say never, but it's usually not successful when it is seen as an institutional mandate to somebody, okay, you must be entrepreneurial. No, I don't think it worked like that. I think if institutions can listen, and I think that's what I'm really saying, is that UBC is listening. And if there's enough interest, and I clearly see there is, and if it comes from the ground up, I think it can happen. So that's what I think that's more. I don't think there's some kind of hand-me-down that any of us can expect from the university. But the fact that the university is willing to provide tailwind is what I think is the critical factor here.
Right. There's another question in the chat that I wanted to touch on as we wind up, and that is the role of philanthropy in these kinds of discoveries and supporting your work. I think each of you probably have your own thoughts on this, so maybe I'll go around the room. Megan, starting with you.
Well, I would just say it's essential. So the little story I told about genetically modifying these Tregs, the initial work we did on that was funded by the BC Transplant Foundation, a small little philanthropic organization with a $20,000 grant. And that was enough to kind of get us going to the next step. And of course, now with that little small amount of money, we've leveraged millions of dollars to fund the research from other sources. So philanthropy has this key spot at just giving sort of like the seed, the spark, the proof of concept that then allows you to take things to much bigger levels.
Right. Sriram, you have a named chair.
I wouldn't be here without the philanthropy. Impossible, absolutely impossible for us to do what we were doing. And that leap of faith that donors have, in this case, through the VGH Foundation, and more recently through a donor who supports work that we do and some others at UBC on COVID-19. I mean, these donors, they make it happen, right? So they're really the heroes in many respects, that they are willing to take the gamble and put the money here. And I feel a deep sense of responsibility that we do the right thing. But without philanthropy, certainly the research that we do would not exist.
Peter, on that note of philanthropy.
Well, yeah, I can just echo what Sriram and Megan have said. I mean, certainly philanthropy plays a pivotal role. Of course, the next stage is really the investment. If you really do want to get something going on outside the university, then you need people that will take risks with their money as well. And maybe that's an area where we do need a bit more support. Or is that in the Bay Area and in Boston? There's some very well-heeled venture capital firms that like to fund things that are in their own backyard, and that's coming here. It's certainly more. There's more of it than there used to be, but we do need that in order to bridge what was the valley of death, or what we referred to a while ago. It certainly that's also a necessary component of the whole ecosystem.
So people have been listening to the three of you for the last hour and a half in this webinar. You've been so generous with your responses and answers to my questions and the questions from our guests, and I want to thank you for that. I want to give each of you an opportunity for a last word. If you were to offer a word of inspiration or a note of, I don't know, focus or direction for those people who are watching the webinar, what would you offer them in terms of thinking about how knowledge is translated and shared in an effort to transform health? Sriram, I'm going to start with you as I ended with you in the first set.
Yeah, thank you, Catherine. Well, first of all, thanks to you and to UBC and the Alumni Association for pulling this together. I mean, I learned a lot in the conversation already. I think we're all, I think what's clear to me is that we all want to make an impact in however small way that we can. So the fact that there's an institution that enables us to do it and has all the bells and whistles that go with it, I mean, what more could you ask for? So I'm happy to be here, and I think that there's many, many decades of great stuff to come at UBC.
Thank you. Megan.
Yeah, I would echo those comments, and really, I feel like the research is at this pivotal moment where, with the collaborations and the connections that are being made now, especially with the support of the Faculty of Medicine and the Translational Academy of Translational Medicine initiative, to really bring people from across the province together in new ways. I guess from my experience, many transformational discoveries happen when you bring somebody like Sriram and myself together who, have to learn to speak each other's language, and then you realize what you can do that you couldn't do otherwise. And I think that the university has incredible potential, and looking forward to working with Sriram and Pieter and other colleagues over the next few years to bring our discoveries to application.
Thank you. Pieter, last word to you.
Oh yeah, just the one thing that's a driver, and I think it's true for all of us, and it was mentioned, I think Sriram mentioned it in terms of getting high impact papers and all the rest of it, but really having, pushing things forward that have a direct impact. Not only, it's nice to think they may have impacts globally, but I do think the local aspect is very important as well. The extent to which we can, people often view universities as being some rarefied place where they do stuff that nobody understands. And I think this is a great opportunity to say, okay, look, some of the work going on in the university is directly affecting your life, and this is so it's a very valuable part of your society. And I do think we need to emphasize that more and bring that home more.
Well, I so appreciate the generosity of all three of you in the response to the questions and to the stories that you share. So thank you very, very much for joining us today. UBC is so fortunate to have you and your big brains and your big hearts. It's great to see scientists sharing a little bit about the feeling side of the ledger, because we don't get to that very often. And for folks like me, that's the joy of this work. So thank you very, very much for taking the time out of your very busy schedules to do this. Thank you as well to our viewers, to the people who helped to make this possible, UBC alumni, to donors, to friends, all of you for joining us in this important discussion. The recording of this webinar will be available through med.ubc.ca/giving/webinar-series. So you can find it. The URL is clearly showing on the screen here, or should be soon. There we go. And you will be able to tap into that and pull this up if you want to. Just real hands up to our three incredible guests today. What an honor, what a humbling experience to speak to the three of them. And I'm just so grateful for having been given this opportunity. So everyone take care. Thank you, science, for so many things. And please support your scientists, doctors, people who work in the medical and health professions around you. They've all made this last year and a half palatable for most of us, and the discoveries are going to keep on coming. So thank you for your time today, and we'll be back to you again as this series, we hope, continues sometime soon. Take good care.
Thank you.
Thank you. Bye-bye.
Bye.